Okay, hi grade 10s. Today we are going to do lesson 4, which is the structure and functioning of the endoplasmic reticulum, the ER, rough ER, and the Golgi body and the ribosomes attached to the ER. Now, we need to join all this together. So at the beginning of the year, we learned about proteins and we know they're made of amino acids. Now, when you eat, maybe you had chicken or fish or cheese or drank some milk, that is made of protein. So when that passes into your digestive system, then there are enzymes in the stomach which start the breakdown of protein, and the small intestine completes the digestion of protein. And then those amino acids, they pass into the bloodstream, and then they get taken to every single cell in your body. So all your cells have got a pool of amino acids that they can use to make your own proteins. So you might remember at the beginning of the year, I always discuss, where are proteins found in the body. So, for example, your stomach, cells lining your stomach, the gastric glands, they're not going to make antibodies. Okay, so every single cell in your body has got all the chromosomes, all the threads of DNA, and on those threads, okay, let's use this. On those threads of DNA, there are regions called genes, and those genes then instruct the cell which proteins to make. So the stomach cells, the lining cells, have all the chromosomes, therefore all the genes, but only the genes for making the enzymes to digest protein will be switched on. The gene for making the antibody enzymes that white blood cells make will not be switched on. All right, so the amino acids that get taken to your cells are used to make proteins, and that process is instructed by the genes on the DNA. So the stomach cell will make enzymes. The pancreas will make enzymes and hormones. The white blood cells will make antibodies to fight viruses. Yeah, we know all about viruses and bacteria. The red blood cells will make hemoglobin to carry oxygen. The cells in your hair, will make keratin to make your hair strong. The glands, like the thyroid gland or the pituitary gland, they will make hormones. So, and even mucus in our saliva is a protein. So the salivary gland cells, the genes for making mucus will be switched on. So that's what we're basically trying to put together. How does the cell know which proteins to make? All right, so let's share a PowerPoint. So you might remember yesterday we were looking at the mitochondria. Okay, so now that's not what we're doing today, and that's not what we're doing today. Yes, we all want to go and sit on the couch and watch ER. Yes, I know the feeling. But today we are going to look at how these cell organelles help our cells to make the proteins using proteins that we ate. All right, so there's just some terminology here. So here's a diagram of a cell, and they've taken out there's part of the nucleus. You can see the nuclear pores, the little red things. So the DNA and the nucleoplasm and the nucleolus would be here. And there you can see the rough ER attached. It's like a maze. It's a system of membranes. Now those membranes, that network of membranes, they are known as cisternae. All right, so it's like a maze going through the cytoplasm so that substances can be transported from the nucleus to the cytoplasm and back. There's links between the nucleus, the control center, and the rest of the cell by means of the endoplasmic reticulum. Endo, inside, plasmic, cytoplasm, reticulum, branching passages. All right, now that ER is continuous with, you can see it's attached to the nucleus, it's near to the nucleus and the cell membrane. But we're going to show how that works just now. And it's all about increasing the surface area in the cell for making proteins and for transport of substances through the cell. All right, now this electron micrograph, you can see here, that diagram. If you imagine taking a section through the ER, using an electron microscope, a transmission electron microscope, this is what you're going to see. There's the cisternae of the ER, and because there are little ribosomes attached, it's known as rough ER, and in between here would be the cytoplasm, and here's the smooth ER. Smooth ER looks more, all over the place, okay, and there are no ribosomes attached. All right. So the comparison between the two types of ER. So rough ER helps our cells to make proteins, and those proteins are assembled with amino acids, at the ribosomes. So the amino acids are brought together in a specific order to make the protein, like hemoglobin, antibodies, etc. But that process, protein synthesis, we actually learn about in grade 12. All right. The ER also transports those proteins throughout the cell. Maybe they need it in another part of the cell. Maybe they need to be secreted from the cell and leave via the cell membrane. All right. The rough ER also makes membranes. So membranes, for cell membrane, for other organelles. But we really need to understand that rough ER is where proteins are synthesized at the ribosomes, and it serves as a transport system for proteins through the cell. Okay, smooth ER, no ribosomes, so it makes lipids. S, okay, smooth makes lipids. So things like oils, think of peanuts, phospholipids, the little lipids that end up as part of our membranes. So, okay, I can't find it now. And steroids, if you think certain hormones in our body like testosterone and estrogen, they are lipid-based hormones. So they've got a protein part, but also a lipid part. And that's where the lipid part is synthesized at the smooth ER. And then obviously the smooth ER also transports those new substances through the cell. And just something out of interest, your liver is a very active gland. It's the largest gland in your body. And it does many, many things. It purifies the blood, it detoxifies the blood. So it would have lots of smooth ER to get rid of, if we take medication, then the medication will do the job at the cells, wherever where the pain or whatever is. And then the breakdown products of that medication will be taken out of the body by the liver. So if you are on medication or if you take drugs, then that's why they check your urine, because they're looking for those breakdown products coming from the liver, because the liver helps to detoxify those chemicals in our body. The same with poisons, okay? That's how they can find out if someone has been poisoned, by looking for the breakdown product, the metabolites in the urine of someone who has been poisoned or is taking drugs or medication, okay? All right, so here you can see a stereo diagram. So that's similar to the first one that I showed you. Okay, the smooth ER, the ribosomes attached to the rough ER. This is just quite a nice one. It almost looks like lasagna. Okay, so you've got the passages of the ER, the rough ER, and there are ribosomes attached. And you can see how it looks like, the tunnels that ants make in the ground. All right, you can see there the little cisternae linking up with each other. That's all the blue part is the rough ER. And here's another diagram. So there's rough ER over there, and there's some smooth ER. All right, now what's quite interesting, the ribosomes is where the proteins are assembled. So if you were able to look at those ribosomes, all these little stringy bits here, those are the actual proteins. So little amino acids are being joined in a very specific sequence, and it becomes the proteins that our cells then use: hemoglobin, antibodies, keratin. And we call those polypeptide chains because they're made up of lots of little amino acids. And then they need to be taken to where they are needed. And that's what the Golgi body does and makes them ready. All right, here's a magnified view of the ER cisternae, and there's the little ribosomes. And ribosomes are actually quite complex. There's a large subunit and a small subunit. We don't even need to know that in matric, but you can see here, there's the ER membrane, there's the subunits, and you can see this little protein chain being made by amino acids being added, and eventually it's released, and now we're going to see, well, what happens now. Right, and that's the job of the Golgi body. So I've got a summary diagram at the end, and the videos will also help you to understand this process, this moving process, okay? So Golgi body or apparatus, it's named after the person who discovered it, okay, in the cell the first time, and they are found in plants and animal cells, eukaryotic cells, near the nucleus, near the endoplasmic reticulum. So there's little membranous sacs, just like the ER is made up of membranous sacs called cisternae. The Golgi body is also made up of membranous sacs. So it reminds me of the Wi-Fi signal. Or I sometimes talk about if you buy pita breads, they come in a plastic bag. There's lots of pita breads, but here you can see how some of them are smaller and then larger and larger. It also reminds me of a lava lamp, if you know what a lava lamp is. So bits break off. You can see here, wait, not quite. There's vesicles that come from the ER. They join, they break off, they join, they break off, they join, they break off, and then eventually they move to wherever in the cell they are needed, or to the cell membrane. So here, these are the cisternae, those little membranous sacs cut across of the Golgi body, and these are vesicles breaking off, and they would contain the protein that has now been packaged and made ready for use by the cell or for secretion from the cell. You'll see in the summary diagram just now. All right, now sometimes in animal cells, when these vesicles break off the Golgi body, they're full of digestive enzymes, and I'll show you what they do a little bit later on. Right, so this is a very active cell because it's got lots of Golgi bodies. Look at all of them, and you can see these little balloon-like structures. Those are the vesicles breaking off with the proteins that have been made inside. And you can see at the edge here, okay, those vesicles actually join with the cell membrane. And say, for example, it is enzymes. Then those enzymes are secreted, and then they can go digest your food in your intestine. Or if they are hormones, then they will pass into the blood and be taken to the part of the body that they are needed. Or white blood cells. White blood cells would have lots of Golgi bodies and ER, and then the Golgi bodies vesicles will break off with those antibody secretions, and then those little vesicles will fuse with the cell membrane, and the antibodies are released into the bloodstream to go and find the germs, the bacteria or the viruses. All right. So this is the kind of diagram that they will discuss on a video. So just to show you here, here's the rough ER. We don't need to know forming phase and maturing phase, but here are the ribosomes. So the proteins are made at the ribosomes. They then travel through the ER. Little vesicles break off with the proteins. They then join with the cisternae of the Golgi body, and then break off and join. They haven't shown everything here. Break off and join, and break off and join, just like a lava lamp. Bits breaking off and joining with the next cisterna. And eventually you can see here these vesicles breaking off, and they would have the mature protein inside. Now, if it's certain enzymes, then those vesicles are known as lysosomes. And we will see what lysosomes do later. And here you can see vesicles breaking off, and they're going to fuse with the cell membrane. So that's when the cell, for example, secretes mucus, or pancreas cells secrete enzymes for digestion in the small intestine, or white blood cells secrete antibodies. All right. We don't need to know this part over here when substances are taken into cells. Okay, so they called this the Golgi complex or Golgi body, and that's where the proteins are matured, and eventually they are released in little vesicles, and now they can go do their job. And there's another diagram. So ER is close to nucleus, and here you can see the little proteins being made at the ribosomes, then they move through the ER, vesicles break off, join with the cisternae of the Golgi body, break off, join, break off, join, break off, join, and eventually exocytosis. I don't expect you to know that word, but that protein is now secreted, like mucus from your salivary glands, etc. All right, so ribosomes, we know quite a bit about them, well, what we need to know. So ribosomes, remember there's a store of RNA in the nucleolus of the nucleus, and that's where ribosomes are assembled, and they're also made of protein, and then they leave the nucleus and they attach to the ER, and that's where they're going to put together those amino acids. It's like workers in a factory, taking little beads and putting them together in a specific sequence to make a specific protein. All right, so they are the cell's protein factories. And there are also free ribosomes in the cytoplasm. Okay, and think of mitochondria. Remember we said they've got their own DNA and ribosomes in the matrix. So that's where they make their proteins. In our cells, they make, in our ER, the ribosomes are attached to the ER, or they're found free in the cytoplasm. All right, so this is now my summary diagram. Normally I give it to you in class, and we color it in and we draw, add things to it. So here's the DNA. Okay, so there would be a gene on the DNA which is going to instruct the cell, it will be switched on, and it will help that cell to make a specific protein, like red blood cell making hemoglobin. All right, so here we can see there are ribosomes attached to the ER, all right, and then there's smooth ER as well, which has no ribosomes, so it makes lipids. Okay, so there we can see the ER. It's made up of interconnecting membranes called cystine, and they transport those newly made proteins around. And you can see here the proteins are being made at the ribosome. There, I've said it over there. And then those proteins go into the ER, and then they pinch off in little vesicles. So those proteins are now in the vesicle. They pinch off the ER, and it moves, that vesicle moves and fuses with the first cisterna of the Golgi body, and then another one, and another vesicle breaks off, and then it rejoins with the next cisterna, and so on and so on. And you can see here now these proteins are drawn in little circles. So it could be a glycoprotein, like the ones that make cell membranes, or certain proteins have to have certain things added, like sugars, and then they have to fold in a specific way. And that's what the Golgi body does. It gets the proteins ready for use in the cell or to be secreted by the cell. So here I've drawn a vesicle breaking off, and this vesicle has got digestive enzymes. Now what it can do is fuse with old mitochondria, and then those substances can be used again, what the mitochondrion was made of. So they help to break down old organelles. The lysosome enzymes help to break down old organelles in the cell. Or if you remember back to grade 9, when amoeba, that little unicelled amoeba, it's living in a pond and it takes up food, but now it's got a food vacuole, it's got to digest it. So a little lysosome vesicle fuses with the food vacuole and digests the organism, and then those nutrients are absorbed into the cytoplasm of the amoeba. So that's one function, okay? Helps to break, lysosomal enzymes help to break down mitochondria, for example, old mitochondria, or in amoeba, it helps to break down the food that the amoeba took in. So then the other function, when the vesicles break off and the proteins in them are for secretion, like hormones secreted or enzymes secreted by the cell for digestion. I've said here, the Golgi body, whoops, where's my mouse gone? The Golgi body is a series of flattened discs called cystine, which package and modify the proteins for use by the cell or transport out of the cell. You can see that vesicle is broken off and it moves to the cell membrane, fuses with the membrane, because remember it's a fluid mosaic, the membrane can change and join and whatever. And then those hormones, it's just a bit cut off over here, the hormones are released or they are secreted from the cell at the cell membrane. That's the story of how ER, Golgi body, and vesicles work together so that your cells can produce, use, or secrete different proteins.